What I'm going to talk about today relates somewhat to my work, but not entirely to my work, and I'll make that clear as we go along. I want to start by mentioning something totally different to begin with. I have been supported by government agencies since 1971, mostly the NIH, and I think many other people in this room have also been supported by the NIH and other government agencies. Over the years, if I make a rough calculation, maybe a penny or two of your taxes, or let's say the approximately 100 million people who pay taxes every year in the US, was devoted to supporting my research. So instead of waiting till the end of the lecture and showing a fancy list of all the people who supported my research and the people involved, I'm simply going to say thank you very much for your support. I appreciate it, even if you're not aware of it. It was very important to me over the years, and thank you. Okay, so I'm going to talk about the origin of life. And contrary to what I usually say at the beginning of lectures to undergraduates here, I'm not going to solicit generally questions from the audience during the lecture, because I'm going to say some outrageous things, some of you may think, and I don't want to get too involved until the end of the lecture to try and answer any questions you have. First of all, one can ask some questions about life itself, and perhaps I should start by giving a very simple definition of what I think life is. That is to say, a living organism should take energy from the environment around it to sustain itself, and it must reproduce itself exactly. That is, a genetic material must be reproduced exactly from one generation to the next. That's my limit of definition. One can add various other things, and I'll probably mention some of them later, but right now, that is my definition. Now, one can ask some questions about life, and I think that some of them, and in your minds perhaps, might require philosophical answers to these questions. Let me state right now that I do not believe there's a philosophy of life, even though I have met people who say they are philosophers of life. I just don't accept that, period. And if you ask me questions about the philosophy of life, I'll refuse to answer them, or I'll grunt. Next question one can ask is, obviously one can think about the scientific origin of life, and we'll talk a little bit about that. I do think about it from time to time, but not very often. But people try and do some experiments in which they try and reproduce some of the basic features that have to do with the origin of life. In fact, Jack Szostak, who I think will be here next week, is going to talk about certain very primitive pockets made of lipids through which molecules can enter, and that's supposed to be has some relevance to the origin of life. And you can do that. And there are many people who do experiments of that kind. But one question that can certainly be asked is, what relation do they have to the origin of life? I mean, you can do an experiment today, and you can get an answer to your experiment, but what does that have to do with what happened, let's say, 4.5 billion years ago? It's not clear that you ever will have a direct answer to that question, but what you will learn may help you think about what may have happened billions of years ago. So I think the people who wish to do those experiments should. And I will say something else about experiments of that kind. That is to say, regardless of what you may think about it, we're all problem solvers. That is, if you're in your kitchen and you drop a kitchen tool or a valuable coin that rolls behind your refrigerator, you're going to wonder how to get it out. Do I have to move the refrigerator? What else do I have to do? But you're going to spend some time thinking about that. And I don't think that's very unusual. We tend to do that no matter what field we're in. We try to solve problems, and this is another problem that we try to look at and think about in creative ways. Now, you can ask yourself, where did life originate? And there are possibly two answers to that. First of all, if you speak to geochemists who study ancient times, they will give you their estimate of what chemicals existed on the earth billions of years ago and whether or not those chemicals can contribute to the origin of life. In fact, that's one possibility, that these chemicals came into existence and somehow, and we'll talk about it, they were organized into molecules that we recognize today. But Francis Crick and Leslie Orgel some years ago, in thinking about this problem, decided that there were some aspects of the chemistry of making essential components of one of the molecules that carry information was too difficult and had to be carried out under very exotic conditions. And they sort of gave up on this idea of life originating on Earth. However, the question is, and so they proposed another solution, which I'll get to in a second, but the question is, if they gave up on Earth, then they must have considered a similar origin of life somewhere else, because this is the only, so to speak, life that we know about. And they thought about that, and they proposed another term, I think the term had already existed, called panspermia. And the idea was that life originated somewhere else in the universe, not in our Earth. In fact, we know that there are meteorites that have landed on Earth that actually came from other planets. Apparently, there are some meteorites that have been identified as coming from Mars. That is to say, there was a tremendous meteorite crash on Mars. Some of the fragments flew out into space and landed on Earth. And you can look at carbonaceous meteorites that occurred in various places on Earth, some, for example, in Antarctica, and analyze them closely. And Sandra Pizzarella, Arizona State University, published a paper in PNAS recently in which she showed a spectrum of hydrocarbons that were found in this meteorite. And there's a tremendous variety of hydrocarbons, some of them seemingly having something to do with the origin of life. However, I feel that's rather a tenuous description of where life came from. It's not apparent that it came from somewhere else. If it did, one must have had one bacterium, supposedly, land on Earth and then generate for millions of generations to ultimately lead to the creatures that we know have evolved since that time. I think, to me, that's very unlikely, and I will simply cite some data recently that shows that, as I said before, the purines and pyrimidine bases can be made quite simply. Okay, so I'm going to show a number of slides, and some of them are old slides, and some of them I will try and indicate where they came from. This slide appeared in the journal Science maybe 10 or 12 years ago, and it was in a review article on the origin of the universe. And let me just go through it very quickly. Right there we have this very planet that's filled with red, indicated that it was very fiery. And supposedly that was the origin of this planet at that time. So that's 4.5 billion years ago. So the time from the present is indicated here in billions of years. Then after a while, this fiery planet slowed down a bit, and I'll get to that in a minute. And we have something with a stable hydrosphere. That is to say, the sky and the ocean had supposedly stable components to it, and we'll have to talk about that. 4.2 billion years ago. Then we had prebiotic chemistry, that is to say, some of the molecules that were important and what we think is important in life are illustrated here, but that doesn't mean anything. So they were present. Then about 4 billion years ago, we had supposedly the pre-RNA world, and you have one molecule splitting into two as it replicates 4, 8, and of course we don't know that this happened, but that is 4 billion years ago. And then we have the RNA world, which I will talk about a little bit. And then we have the first DNA protein life, and we don't know how this happened yet, but anyway, we'll say that this happened. And then we have the diversification of life. So here we have a simple evolutionary tree that's just indicated here. It doesn't matter what's shown, from 3.6 billion years ago to the present. So that's a very broad description of what went on. Now, I think it's important to ask some very simple questions. And these you might think are trivial questions. In fact, this slide was made about 8 years ago when I had to deliver a lecture to a number of outstanding high school students. And this is the questions that one asked. So this would be the stable hydrosphere. What was the nature of the oceans? Were they all water? What was the chemical nature of the oceans? What particular chemical compounds were in the oceans at that time? Were they all oxygenated compounds? We don't know. We have to know these things. What was the nature of the sky? How much oxygen was in the sky? Or methane? Or carbon dioxide? This is very important in terms of understanding the metabolism of what we think is the beginning of life. Then the temperature is also very important. Were the oceans at, let's say, 90 degrees centigrade, which seems unlikely during the supposedly stable hydrosphere? Or were they down around 37 degrees, which is our body temperature? And how about electrical discharge? Because we know that electrical discharge, in terms of lightning perhaps, can affect various biological reactions. So we're really thinking here about ultraviolet radiation, which we know is important in stimulating many chemical reactions that we're interested in. And finally, the time. How much time can we spend on thinking about the original chemical world in terms of kinetics we understand about various reactions? Now, this slide is from a paper that existed in Nature that came out last November, I think. It was on the appearance of various fossils, and that's not important right now. The important part of this is it simply displays some things that I'm interested in. So here we have time from billions of years ago, 4, 3 to 1 billion years ago, and this is the ocean. Originally, it was an anoxic ocean, that is to say, very little oxygen in the ocean at all. And it became a sulfidic ocean, in which there are many sulfate compounds and oxygen in combination with sulfur in the oceans. And this allowed for many more things to take place, and finally became an oxic ocean, or one in which the compounds were essentially compounds with oxygen in it, and could be very fertile in promoting life. And in the sky we have, for example, this is the oxygen level. We have, first of all, very little oxygen at the beginning, and when the ocean turns sulfidic, we have the great event here of some particular disaster in terms of the fossils that existed, and also the proliferation of a great number of fossils. And then that went up for a while. So this is, let's say, before you make organisms that have nuclei in them. And I should say that many of you, I recognize easily, are scientists, and you will understand exactly what I'm saying. And you might even get bored by what I'm saying. And I don't mind if you leave. And others among you are not scientists, and you may not understand what I'm saying, But that doesn't matter. Don't worry about the scientific details. The general things I'm saying are more important. Okay, so here we have bacteria before we have eukaryotes, and then eukaryotes start coming in. And around this time we have, and this paper was about these Gabon fossils, which apparently have something like nuclei in them. And then that goes up until we form the oxic atmosphere, and we have a whole series of other events occurring right here. In fact, right here we have the oldest unequivocal animal fossils. So that's very recent, only about, let's say, 800 million years ago. Now, Graham Cairn Smith, some years ago, proposed a theory in which he suggested that clays were very important in terms of the origin of different simple organisms. And I'll try and describe this very briefly, because it's not apparent to me that it's that relevant. He said there can be, for example, two kinds of clays. You can have clays in which there are crystals, let's say, that are all pointed in this direction, or clays that are crystals that are all pointed in this direction, like that. I don't disagree with that. But then he said that the other smaller molecules that are absorbed on these clays can be specifically fractionated in some ways. And we know that in living organisms, amino acid residues, which are part of proteins, make up one particular form of molecules that reflect light in a certain fashion, or they reflect polarized light in a certain fashion. And Cairn Smith said that, well, in these clays, apparently the amino acid residues, which normally are a mixture of, let's say, D and L amino acid residues, it doesn't matter what they are, they're both of them, they can be fractionated, and one of the forms here can remain on the clays and participate in further reactions that have some particular direction to them. I would say that the direction has something to do with life, and the other form is lost completely. Okay, that was a theory that was popular with some people, and I don't think it has any real seriousness behind it at all. So I leave that at that point. It is still not clear to us how we do get separations of D and L amino acids, or for example, D-riboses from other ribose forms, but all that does happen, and those are important problems that we have to solve. Okay, so I'm going to start talking now about molecules that I think are important in terms of the genetic continuity of living organisms. Now, it is possible that proteins have information in them. In fact, you can designate proteins so that different points on them can be represented as a bit of information. But from what I've said now, what I will say, we don't have to pay attention to that, because proteins cannot reproduce themselves in any particular way. You cannot take the information in proteins and make more proteins with exactly the same information from them, only with the original proteins. And in fact, we think that RNA and DNA are probably the basic genetic informational molecules. And if you look at DNA, for example, we know that inside cells, aside from one or two viruses, DNA is usually a double-stranded helix, so we got two strands of a helix winding around each other, and it doesn't exist in any other form. You could say that during replication of DNA that there's a small region which is denatured, opened up, but it's only done that with proteins, so we'll forget about that. But RNA is quite different. RNA exists as a single-stranded molecule. That is, these are very long molecules, okay? And they are made up of repeating subunits. And in one of your cells, you have approximately 1 meter of DNA stretched out in one form, if you stretch out all the DNA. Now, Since RNA can be single-stranded, it means various things. One is that it can attain any shape it wants to in solution. That is, there's nothing to inhibit RNA from taking various shapes inside solution. And RNA has information in the same way that DNA does. It also, as I said, has a repeated sequence of subunits, and that's about it. And so that's all we have to know at the moment. Now, let's just look at the chemistry of RNA. And this is probably more than you want to know about the chemistry of RNA, but these are two of the subunits of RNA. This is the ribose ring, a sugar ring, here and here. And these are bases here and here. This is a purine base, adenine or guanine, or a pyrimidine base, cytosine or uracil. And the sugar, as I said, is here. Then there's a phosphate backbone here and here. Now, you can make various derivatives of RNA, which I'll mention probably a little later. And once the phosphate backbone is substituted by different kinds of phosphate backbones, the sugars can be substituted by different sugars, and even the bases can be substituted by these bases here and here. Supposedly all of them can participate in hydrogen bonding. The point is that although there is an RNA structure that we know about today, it's possible that at one point there were structures that looked like RNA which may have contained some of these other molecules. And for example, I think it's Eschenmoser made an RNA with hexoses, 6-member sugars here, and they participate in hydrogen bonding, but they didn't correspond at all to the melting characteristics that we know of RNA. Let us say if you have 2 strands of RNA which hydrogen bonded to each other, and I'll go into that in a little while, you can supposedly melt them by heating them, but the hexoses didn't do that properly. And DNA, just to indicate properly in terms of the chemistry, DNA only has a hydrogen group here. It doesn't have the hydroxyl group, just has a hydrogen group. So DNA is called deoxyribo, which means no oxy in this position. Okay, now this is an old slide made by Hugh Robertson many years ago. Unfortunately, in my view, very unfortunately, he died a few years ago. And this is to show some of the ways in which you recognize pieces of RNA. So this could be just a long piece of RNA, and the letters we use here simply indicate the abbreviation for the bases in the RNA. So we have A, C, U, and G, which are the 4 bases, and you can have them specified along here, and maybe this is a signal of some kind, but I won't even say what it's a signal for. Then we have secondary structure. By secondary structure, I mean a way in which the pieces of RNA fold together to make what looks like a double-stranded structure. So, for example, here you have this sequence right here, and then you have, in terms of Watson-Crick hydrogen bonding, a complementary sequence right here, so that U pairs with A, C, G, AU, AU, GC, GC. And you would have all this hydrogen bonded, which means it stays together from, let's say, 30 degrees up to maybe 50 degrees when you can melt it. But that's very important, and this kind of structure will, not necessarily this length, but this kind of structure will form sort of automatically. In any piece of RNA, let's say that's 100 nucleotides long, you will have self-complementary regions in there, and they will form these double-stranded structures. That's very important, as we shall see later. And then you can have various other signals here. For example, here you can have structure. This is a particular structure that might be interesting. This would be double-stranded and some sequence out here, et cetera. So this is all for people who know something about nucleic acids. Very simple, but it's important just to know that RNA does all these different things. Okay, so this is the iconic central dogma, as Francis Crick called it when he enunciated it in the late 50s and early 60s. And it represents the transfer of information as we understand it today. And that is to say, and this is not the whole scene, but this is the central part of it, You have DNA with genetic information. From that you can make RNA, and from RNA you can make DNA. And RNA then goes to the ribosomes, and it makes proteins, which have structure and biochemical catalysis, okay? So all biochemical catalysis is carried out by proteins. Now, when this was enunciated, and I should say revered, one has to assume that there were proteins at all points in this case, and everything that I just mentioned was carried out by proteins. So that there is a protein enzyme that replicates DNA polymerase, replicates that. There's another protein enzyme called RNA polymerase, which copies DNA and makes RNA from it. And there's a reverse transcriptase enzyme, that all made of protein, that copies RNA into pieces of DNA. And the ribosomes are made of RNA and protein, and we'll certainly talk about them later. But the point is, this only works if you have proteins present to carry out all these reactions. In 1967, Carl Woese wrote a book about evolution, and Crick and Orgel separately wrote papers about evolution. And all three of these people concluded that they had no idea how life originated, because they had no idea how any of this happened in the absence of protein, and they made the assumption that there was no protein. There could have been DNA and RNA at that time, but there was no protein. So this is a major puzzle which Crick and Orgel and Woese had to solve. Now, here is another view, which first represents exactly what I just said. You have DNA going to RNA, but there's no future here in terms of the beginning of life, because there are no proteins. Then in 1982 and 1983, some discoveries were made showing that RNA could have chemical catalysis, okay? So now we had RNA that had information and biochemical catalysis, and now you have possible future, because you could say that you made small organisms that had RNA to carry information, and RNA could carry out biochemical catalysis. Now, what was done at that time was something that scientists invariably do. You have this, and at that time it was known that there were only two reactions carried out by RNA, and they were very simple reactions in which RNA cut other pieces of RNA. That doesn't matter. The idea is once you have a reaction that does something, you can say, okay, that molecule does that, and therefore we can assume that molecule can do any kind of enzymatic function. It's a very broad statement, and it's always done by scientists when they uncover new properties of a particular category of molecules. It's done by not just biologists, but by physicists too. Then in '86, Wally Gilbert from Harvard wrote a small opinion piece in Nature, and he said, well, all this is fine. He said, but in fact, you don't need DNA at all at this point. Why have DNA present? You don't need it. All you need is RNA. If RNA has information and it can carry out biochemical catalysis, say one thing it could do is replicate itself and do things like that, and do lots of other things which we don't know about at the moment, then we'll just call this the RNA world. And that phrase, the RNA world, has pretty much guided us in our theories about ancient organisms up to this point. So that leads to a possible future in terms of the origin of life. Now, I should say, even though I've said that Wally suggested this, and that's what we use, the fact is we don't know whether or not DNA was around or not. We just don't know. We have no knowledge of it. And secondly, while we say it's the RNA world could have consisted of many derivatives of RNA, like the ones I mentioned earlier on, so that it's not exactly the RNA world we see at this particular time. Now, there were some people who were stimulated to find whether or not RNA could have other enzymatic reactions. And Larry Gold at the University of Colorado invented a technique which enabled people to do this. I'm going to show a slide from one of his early papers, but don't pay much attention to it. I'll just point out what's important, I hope. Okay. You started with a randomized template, a randomized piece of DNA, or a piece of DNA that could be made into RNA. So you might have a piece of RNA that's 100 nucleotides long, and by randomized, we mean at every point in that piece, at every place where there's a base sequence, you make this by machine, and you put in all 4 bases at every point. Okay, so you might have A, C, C, G, etc. Then you will have G, C, C, G, G. Then you have C, G, G, C, C. You have U, G, G, C, C. And that will occur at every point along the way. And in that sense, you could very easily, you can prepare a half mil of a solution, half milliliter, it's a very small amount of solution, with, let's say, easily 1 trillion or more different sequences available. So that is very important, and it's a very useful tool. So we have this population, and then we do a selection on it. That is to say, if you have a particular particular enzymatic reaction you're looking at, let's say that takes methyl groups, CH3 groups, and adds them to something else, you can attach the CH3 group to a solid support and put it on a column, just a linear representation of that stuff in a solid support, and pass the RNA material you have over it, and anything that binds rapidly to that methyl group will stick to the column. Everything else just gets washed through. And then you can, after you've done that, you can raise the salt conditions to some extent, and what's left bound to the column will be washed through. And you can do that many times and make the elution properties from the column more and more stringent. And finally, you can wind up with one molecule of RNA out of the many trillions of them that, in fact, seems to have the right properties and could be what you thought of as having the proper enzymatic properties. So that was done with many different enzymatic reactions, even though I haven't described it well. And we have here a slide that was made, except for the last 2 lines, about 10 or 15 years ago, in which we have about 15 different enzymes here, all of them important for life as we know it in bacteria today. And these were all isolated by various people over the ensuing years from Gold's original discovery of this method, the SELEX method. And from that time, there are probably 10 more isolated. So people, let's say, isolated about 25 different enzyme functions, all of which were due to one particular piece of RNA. And I should say, with that particular one piece of RNA, it's very easy to replicate that many, many times. So there's no problem dealing with one molecule of RNA. We're dealing with millions and millions of molecules of RNA that can do this reaction. So we have about 25 of these that can happen. And people got tired of doing that, and I don't blame them. It's very useful to have done it, and it was instructive. Now, in addition to that, various molecules, various pieces of RNA were found with small molecules attached to them. For example, coenzyme A, which is an enzyme cofactor, and that doesn't seem to be too difficult to do. And pyrimidine and purine synthesis has occurred. Now, this is a very important goal that was reached by John Sutherland, who is now at the MRC Lab of Molecular Biology in Cambridge, England. He has succeeded in making purine and pyrimidine subunits of RNA by doing it in essentially large phosphate-controlled, essentially, reactions, and he can do it essentially in 2 or 3 days, possibly even a shorter time than that. This is the reactions that Crick and Orgel were found to be virtually impossible to do easily, especially Orgel, who was a very good chemist, and he gave up because of that. But Sutherland has done this, and I We'll do nothing but congratulate him for what he has done. I think it's very important. So let's shift from a long time ago to today. And here we have examples of catalytic RNA today. And by the way, I should say that none of those enzymes that I just showed you a minute ago have been found today. They don't exist in nature as far as we know. Or that is to say, we haven't been clever enough to find them in nature. And one can object to that in two ways. One saying we never had, for example, a collection of 1 trillion different molecules of RNA to work with, which is most probably true. And we don't know what time it would take to sample such a large number of molecules to see if they had the right enzymatic conditions. So while people have found those enzymatic conditions, we have no idea whether or not they exist today. In fact, so far as we know, they don't. Okay, so here are various examples. There's the RNA subunit of an enzyme called RNase-P, which cuts other pieces of RNA. There's group 1 and group 2 introns. I won't explain what an intron is right now. I'll come to that in a little while. But group 1 introns, founded by Tom Cech, also cut themselves in a certain way. Plant viroid RNA, and I'll talk about that just briefly. Plant viroid RNA, single-stranded RNA, which is a circle. It's a closed circle. And it doesn't code for any proteins. It's usually about 350 nucleotides long. And these are extremely important as plant pathogens and as dangers to the commercial production of plants. Virtually every plant that you can think of, that let's say fruit you can see in a market and plants come from that, has a viroid which will attack the plants. And in fact, you can take some viroid RNA on your fingertips and rub it into the leaf of a plant, and 3 weeks later, 4 weeks later, the plant is dead. That includes coconut palms. And the reason that your fingertips, which actually have nucleases in them that degrade RNA, don't attack this RNA is that the RNA is a closed circle. So it's not easily attacked by these nucleases. Then we have hepatitis delta RNA, which is very similar to plant viroid RNAs. This is 1,600 nucleotides long, again, a closed circle. It codes for one protein, and it's important as a human pathogen. If you have hepatitis B virus in your bloodstream, you get sick a little bit. Sometimes, occasionally, that will transform into a cancer, but you do get sick from hepatitis B virus. However, if you have hepatitis B virus and hepatitis delta RNA, you die within 3 weeks. And the mechanism by which this happens is not fully understood at the moment. So I'm just saying this as a piece of information that might be interested. Then there's a fungal virus, very similar to these viruses, ribosomal RNA elongation site. As many of you know, ribosomes were crystallized by Tom Stites, other people recently, and if you draw a sphere around the place in the ribosomes where 2 amino acid residues get joined together to mark the continuation of a polypeptide chain, there is no protein in that sphere of about 20 angstroms around that point. So the statement is made then that peptidyltransferase in ribosomal RNA is an RNA enzyme by itself. And I should indicate what an enzyme is. I'll show you a slide of that shortly. But the fact is, an enzyme is something that carries out a reaction and is not disturbed by itself during the reaction. So for example, in the old days when lead was in gasoline, lead was a catalyst that promoted the explosion of the gasoline mixture, and lead did not change during this reaction. So in this case, this does not change during the reaction. In this case, the RNase pRNA does not change during the reaction. And then we have splicing RNAs. You can make a complex of certain splicing RNAs which are involved in tailoring molecules with introns in them, and I won't talk any more about them. You can make a reaction that looks as if it's a natural reaction. This was done by Jim Manley and a woman who worked with him, and it seems to be a reaction that doesn't require any protein. And finally, we have riboswitches, they are not really enzymes, but these have been discovered mainly by Ron Breaker here at Yale. These are pieces of RNA that can bind other very small molecules, let's say vitamins or amino acid residues, and they are involved in regulating the production of other things that the RNA encodes. I won't say anything more about that, but the riboswitches are important in these ways, and you could in some way include them in this particular list. Okay, so in biochemical terms, an enzyme is a catalyst that works in a chemical sense again and again on other class of molecules, which are called substrates, and it is unchanged by the reaction. And so, for example, there is an enzyme called ribonuclease P, and its substrate, among others, are tRNA precursors, and I'll talk about this a little bit. So here, for example, is a transfer RNA precursor, and this is the way a piece of RNA can be drawn on paper. These dots indicate hydrogen bonds here, 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 and here. They're unimportant for us right now. The beginning of the transfer RNA, which is important in protein synthesis, is at this position, which is marked by 1 here. We'll forget about this for a moment. And the RNA subunit of ribonuclease P cuts this molecule right at this point here and gets rid of all these upstream sequences. This is important because when this molecule is copied from DNA, all these nucleotides are included. So ribonuclease P cuts right here. Now, there are about 60 different tRNA precursor molecules in every cell type because there are about 60 different iso-accepting species of tRNA. If you look at the sequences around this site, they're not identical at all. So what else controls the specificity of this enzyme for this substrate? Over several years of work, it was found that the important thing is simply this apparently double-stranded region here and this single-stranded region here. So the junction between a single-stranded region and a double-stranded region here is what controls the specificity of this particular enzyme. And as I said, the identity of these particular nucleotides is not very important. In fact, you can only have 3 or 4 of these base pairs here and 1 nucleotide here, and the enzyme will still work. So what else is happening? And that, by the way, has been shown by many years of extensive experimental data. This is a speculation based on that data which I just mentioned, and it has something to do with the origin of life, or it doesn't have anything to do with the origin of life. It's just pure thought floating in the ether, so to speak. You can have a large piece of RNA to begin with, okay? And in this large piece of RNA, we have these supposed hairpins, that is, double-stranded regions with loops. And these are not uncommon at all. You have them frequently in large pieces of RNA. And then you have this particular RNA subunit of RNase P, which at that time would just be the RNA subunit by itself. And it cuts this molecule right here at the beginning of this hairpin, and right here at the beginning of this hairpin, and right here at the beginning of this hairpin. So this is speculation. It simply says that this particular enzyme has the ability to cut this molecule in several different places. And molecules such as this, we presume, must have existed many years ago when life was extremely simple. But once you cut this into other pieces, these other pieces can have different functions, as we know. So, for example, you can take this function here, and in fact, you can add to that CCA, which we don't have to talk about, and you can add to that, in fact, an amino acid, which can then function in protein synthesis. And it has been used by Paul Schimmel in experiments to look at protein synthesis. And ultimately, although I will not say this is a strong argument by any chance, you can get duplication of this loop, and then finally a duplication of the loop again, or a similar loop, and you get something that looks like a transfer RNA molecule. So the specificity of this very ancient enzyme can in fact be linked to something that was very important a long time ago. But of course, we don't know anything about that. I should say that when you talk about RNA, things like this, and the ability of RNA to replicate itself, if it did replicate itself, we know today, for example, that when RNA is replicated, mutations occur in it fairly frequently. By that, I mean at the ratio of 10 to the minus fifth or so per rounds of replication. Whereas normally with DNA, it doesn't occur to the replication until ratios of 10 to the minus 7 or 10 to the minus 8th. But the point is RNA can mutate even when replicated by an enzyme. But the other point is that DNA and RNA are chemicals. And I have to say that most students I speak to don't understand that. They see DNA and RNA as diagrams on a page. They're not aware that they're actual chemicals, and they obey all the laws of chemistry. And from that, we know that the base pairings in DNA and RNA will change from time to time. So even without enzyme replication, you will have mutations in, let's say, RNA or DNA that ultimately will lead to evolution of whatever genetic material you're talking about. So, sorry, that's a little aside, but let's go on. This is a diagram of the group 1 intron that had been isolated by Tom Cech. Now, if this is an RNA molecule, it starts here, it goes here, and this zigzag line is part of it, and it ends here, okay? The point is, for this to be functional, as we know it is in this case, is this part here has to be joined to this part here. And these are called exons, doesn't matter. And this is called an intron, so we have to have ways of getting rid of this intron. And Cech found that a molecule that he was looking at this way, which is a precursor to a ribosomal RNA, could in fact delete this particular molecule from this configuration and join this part to this part here. So he made this, although it certainly didn't have this little circle in it here. It just became a line like that, plus the extra intron at this point. So that was also a very important finding, that RNA is catalytic. Okay, so I think I have to move along here. Now I'm going to show another example of the central dogma as we understand it today. And this comes from another review article that I think appeared either in Molecular Cell or Cell. Okay, this is tremendously complicated, but in fact it's not complicated at all. And this is what we understand goes on right now. Here we have DNA, or chromatin, which is the condition in which DNA exists in higher cells. That is the chromosomes with protein attached to the DNA. And there are small molecules called primer RNA, which helps in the replication of DNA. And we have telomeres, which are the ends of pieces of chromosomes in higher cells, not in bacteria. And there's an enzyme called telomerase, which replicates its DNA. But that's not important. The point is you have DNA, and it goes into make RNA, and part of the RNA is made as messenger RNA, which goes to ribosomes and make proteins. Okay, but what's all this jazz here? Many different kinds of RNA are appropriate, or let's say, listed on this slide. And this is very important. And I would say, and I'm going to be absolutely patriotic in this sense to RNA. If you want to study something interesting for the next 10 years, study RNA. You will get a good job, and you'll find out interesting things. You can study other things too, but this is more important. Okay, so we have RNA. You have RNase P, which cuts transfer RNA, tmRNAs, ribosomal RNA in bacteria. I won't talk about these. 7SL RNA, which is in eukaryotes. You have SNO RNAs, which cut ribosomal RNA, snRNAs, and then we have miRNAs, microRNAs, siRNAs, and other non-coding RNAs here. And I'm going to talk about these three briefly. And of course, aside from that, we have messenger RNAs, which, as I said, make proteins. But all these other things do carry out different functions inside cells, and they're very important. In fact, these three are so important that it's worth talking about them a little bit, because it indicates that RNA is extremely important in controlling development and differentiation. Okay, we have non-coding RNAs, thousands of nucleotides long. They do not code for proteins, and for most of them, the functions are not known. Now, one of them is known. It's called the exist RNA. It's a few thousand nucleotides long. It's transcribed from one of the X chromosomes in female cells. And in humans, we know that females have two X chromosomes. Males have only one. But one of the two X chromosomes in females is inactivated. It doesn't show any function at all. That's after a few rounds of duplication of embryos. So one of them is inactivated. And the same thing is true, for example, in mice and Drosophila. And Jeannie Lee at the Harvard Med School has been studying this for several years, and I have to say that she's done an absolute marvelous job on this. She's done the biochemistry and genetics of exist RNA. She's looked at various parts of RNA. She's determined that the exist RNA or parts of it control the number of copies of xRNA of the X chromosome and various other things too. So I have nothing but admiration for her. Hot air RNA has been studied by John Rinn, who's at the Broad Institute in Boston, involved in oncogene expression. And there are many other RNAs involved with epigenetic effects, that is, methylation of DNA, histone positioning. So I would say maybe there's another 4 or 5 large non-coding RNAs have been found for which some function is known, but these occur very frequently in higher organisms, and we don't know most of their functions yet. Now, we also have miRNAs, microRNAs. These are small RNAs. They can be up to a few hundred nucleotides long, but ultimately they're processed into something about 22 nucleotides long, although some of them can be somewhat larger than that. They were originally found in Caenorhabditis elegans, a small worm, and it was found that they controlled certain developmental effects in that worm. And it was soon found that one of them, for example, was found also in human cells, and now the same thing has been found about several of them. And it's absolutely clear that the microRNAs control differentiation and development, very important aspects of it in higher organisms. So we have now 2 RNAs, non-coding RNAs, which are extremely important, as I said, and the microRNAs, and there are many microRNAs in human and mouse cells, Drosophila, Caenorhabditis, and they control several different differentiation effects. And we have RNA inactivation, in which you make large RNAs, a few hundred nucleotides long, that are double-stranded, and they can be processed into smaller RNAs. By processed, I mean there are certain enzyme complexes in these cells which will cut down these larger RNAs into smaller ones. Many of them are microRNAs, and they can function in inactivating or activating some genes. In bacteria, there are also some small RNAs, and I'm not going to go through all of this at the moment, but these small RNAs, let's say between 50 and 200 nucleotides long, are responsive to stresses that you put on the bacteria. For example, by that, you have bacteria generally growing at 37 degrees. You can grow them, let's say, at 25 degrees in some cases, and the bacteria have to adapt to the cold stress. Or you can have them grow at 42 degrees. They have to adapt to the hot stress. You can change the pH. That is to say, you can make the medium in which they're growing very acidic or very basic, and some of these RNAs will appear. So these are stress responses, and I think in many ways they're not totally different from the microRNAs we see in higher organisms. Now, we're discovering new genes, especially, let's say, among, I wouldn't say genes because genes generally mean coding for proteins, new mutations to aid in providing basic information. But this has to do with, let's say, the microRNAs and the non-coding RNAs. And at the molecular level, we essentially use everything inside cells. Many of the new RNAs we found, especially the non-coding RNAs, are transcribed from regions in cells that we thought were non-functional. It could have been junk DNA, or there are regions in the non-sense strand in protein RNAs. Usually we have a double-stranded piece of DNA that codes for a particular protein. Then the other strand could code for another protein. And that's what we found now. And it is important for us to understand the function of these additional RNAs, and in fact, the RNAs that are made from supposedly junk regions. So I think that's terribly important at the moment. What I'm saying here is that effectively, you shouldn't neglect anything. If you're studying molecular biology of information transfer in cells, nothing is immaterial, nothing's unimportant. Everything that you're looking at is important, and it's worth studying. Okay, I think I only have a couple of slides left. This is about the evolution of the RNA subunit of RNase-P, and I'll talk about that in a second. I just want to recapitulate for a second. I wound up saying that it's totally unclear how life originated, or virtually totally. And then it seemed as if it was more or less obvious that it had to start with RNA. And then I've talked about RNA since then as being important in one way or another. And the more I've talked about it, the more important it has become up until where we are today. And just as a coda, I'll mention this is an example of evolution and one of its problems, or one of its many problems that one can present today. In this case, in bacteria, this enzyme has one RNA subunit and one protein subunit. And its catalytic activity is, let's say, we can define as between 1 and 10. And archaea, which is another kingdom of supposed bacteria defined by Carl Woese, very important. There's an RNA subunit of the enzyme, and there are 4 protein subunits. And the amount of catalytic activity is between 10 to the minus 1 and 10 to the minus 2 compared to this. In eukaryotes, which are essentially people like us, or yeast, there are 9 to 10 protein subunits. And the amount of catalytic activity in the RNA by itself is 10 to the minus 5 compared to this. So if you draw an evolutionary tree, this is sort of the beginning, this is almost at the beginning, and this is much later on. Now we see as evolution goes on that the amount of catalytic activity having to do with the RNA subunit appears to decrease, or that's the way we measure it, and the amount of proteins is increased. So the question is, how did this happen? Do the proteins actually have catalytic activity by themselves, or are they simply enhancing the RNA subunit, which I think is most likely what happens in this particular case? And that's a problem which should be asked in these days, and I think there are people working on it, and I hope that will remain the case. Lastly, there's one other slide that I made up yesterday, which I think is worth mentioning. Don't laugh. Okay, we started with the RNA world, and then for a while we talked as if there was a protein world today. And all the enzymes which are important in replication of nucleic acids and many other enzymatic reactions are made up of protein. But I think in the last few years, we can change that. And again, I'm a partisan in this sense. I'm a strong partisan. We still have the RNA world because RNA clearly is extremely important in regulating, controlling genes that have something to do with development. And we have a protein world. Of course, this is in smaller levels compared to RNA at the moment. But that's just my partisanship showing through. Finally, we have a question mark. And that is the next description of our world. I have no idea what that will be, and if anybody has a reasonable suggestion, I'm willing to listen. Thank you. Okay, questions? He's coming with a microphone.
Thank you. Very simple question. Of people who work in labs know that you have to take so many extra precautions working with RNA because it's so easily degraded, whereas you can isolate DNA out of anything without the precautions. Can you give any little insight about if the RNA was the primitive molecule, and also what might be the mechanism for wanting to get rid of the RNA really quickly in in a developmental context if you don't want it around? Like, why does it degrade so rapidly? Any?
Why are we getting rid of RNA? Why do you think we're getting rid of RNA? I don't think we're getting rid of RNA. I don't think we're getting rid of RNA, and other than that, I don't have a reasonable answer to your question. Okay. Down here.
Oh, okay.
No, I'm just going to.
Thank you, Dr. Altman. So you mentioned in the beginning that you thought the building blocks of life was something that has DNA and can replicate itself exactly. So I was wondering if you consider viruses life, and whether or not your definition of life can be also extrapolated onto other planets, because given your ingredients, that could potentially happen. And with proteins being the basic building blocks of life, would you see DNA also being used as encoding protein production on other planets as well?
Okay, I don't believe viruses are organisms of life as I define them, because viruses cannot replicate by themselves. They only replicate inside other cells, right? And I don't know what happens on other planets. I literally have no idea.
You mentioned that proteins are not self-replicating, if I understood you correctly.
Are not self-replicating, yes.
Correct. And how do the prions fit into that picture?
Prions are proteins that are thought to be involved in scrapie and other such diseases. And it had been thought for some time that they did self-replicate, but they don't self-replicate. If you read papers by, you don't have to read them, by Charles Weissman or Prusiner, you will find that the prions are different in conformation, in their structure and solution. And the normal prion gene makes normal prions with the right conformation. If they're put in solution with a prion that comes from a diseased person, ultimately the diseased prion, which is in a different conformation, will bind to some of the normal proteins and induce them to become abnormal in structure too.
So what's being replicated is the structure, not the molecule. It's a conformation.
I wouldn't say it's being replicated, but in fact, the unusual structure gets transmitted in some way, but it's not part of the genetic material. There's one down here. Right here. Oh, sorry, yes.
Okay, thanks. A question which is regarded to your comment that you're very patriotic about the RNA. Well, in the scientific community which is working on these issues, would you say that this, or does that play a role? I mean, are there people who are DNA patriotic and focused to try to defend the DNA. Is ideologic playing a role in the scientific community in these matters? I'm really interested in that. I mean, thanks.
Could you repeat that last sentence you said?
Does ideologic thinking plays a role?
Oh, yes, it's purely emotional, completely emotional. I'm just trying to generate more support for RNA. That's all. And probably there are a lot of people here who would disagree with me about that.
You talked about how important RNA as an enzyme was in simpler forms of life. Why would you say, as an RNA patriot, why would you say that the proteins take over more and more of the enzymes if RNA is sufficient?
Well, that's a very good question because it brings up two things. One is, first, were there so many enzymes associated with RNA to begin with, which we can say is possible, but we don't really know it's the case. The second question is, when did proteins start being made? And they had to be made at some point. And there are some theories now about how RNA could begin to make simple proteins. And then the question is, why did proteins turn out to be so much better at these functions compared to RNA? Now, that's not entirely true because some proteins are clearly better. There is no RNA form that carries out a lot of these functions. But some of the functions are still carried out by RNA themselves, like RNase-P, and which is a very important enzyme. It's essential in all cells. It does all kinds of interesting things. And for example, in the ribosome, where supposedly the peptidyltransferase is critical. So I have no real answers to your questions, but it's important to consider what still remains from the RNA world and what has changed. And we don't know how what has changed happened.
You talked about synthesis of bases and sugars. How do you synthesize RNA?
How do we make RNA?
I mean in prebiotic
Yes.
world.
Yeah. That's exactly what John Sutherland is working on at the moment. And I say that having heard him speak about a month ago at a meeting. He's clearly intent on making long pieces of RNA from these purines and pyrimidines he's able to make. And he makes the whole nucleotide, the sugars with the bases attached to them. So he just has to figure out how to put them together to make long pieces of RNA, and that's what he's working on. I can't tell you anything more about that.
How about Dr. Ferris' work on?
How about whom?
Dr. Ferris?
I'm sorry, I don't quite understand you.
So he was making RNA on clays.
On clay?
Yeah.
Oh, yeah.
So I thought you had a...
No, I don't put much attention to those particular experiments.
Reason?
Well, we can talk about that privately.
Sid, all of this requires energy. Something moves. Are we looking at a phosphate world eventually?
Are we looking at a toxic what?
A phosphate.
Oh, yes. Obviously, that has to be important in terms of making the nucleotides, and John Sutherland admits it's very important. One of the steps that he uses involves UV radiation. And I asked him why he did that. And he said that's because Leslie always did it. Leslie Orgel did it. And so that supplies a certain amount of radiation, but we're only talking about nucleotides right now. And you can argue about some of the other sources of energy to do other things. There are lots of geochemists who feel that iron sulfide complexes, which are present in many proteins, are very important too. But we still have to wait and see what comes out from these experiments.